This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 997, recorded on March 31st, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hello there, Vincent. We have 81 degrees Fahrenheit, sunny, windy, all good.
Here in New York City, it's 10 degrees Celsius and windy also. It's still cold. Overnight it freezes here still. The bird baths are frozen in the morning. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 45 Fahrenheit, 7 Celsius, and it's gone overcast today, and we're supposed to get rain into Saturday, so kind of typical getting into mud season here in Western Mass.
997, we are 3 episodes away from 1,000. I think I have a few seats left, folks. We had some cancellations, so if you're interested, you could still get in. Just send me an email, vincent@microbe.tv, and I'll send you the details. Yeah, a few people canceled, which is too bad for them. They're going to miss out. Our guest today is from Boston University School of Medicine, Mohsan Saeed. Welcome to TWiV.
Well, thank you for having me, Vincent. Here in Boston, it is 7 degrees centigrade, and it is partly cloudy. I think it's going to rain soon, probably from 6 p.m.
You'll get my weather about an hour later.
Yeah. All right. Yeah, I think
it's the only thing that happens first in Western Mass and later in Boston.
Yeah. Cool. Mohsan is sitting. Are you in the same building as the Needle? Is that where you are?
Exactly. So this is National Emerging Infectious Diseases Laboratories, called Needle. Should be called NIDLE, but people call it Needle.
I think that's because of our episode title, episode 200.
Needle.
Threading the needle, and it's forever going to be the needle after that.
Exactly.
Oh, so what was that? How many years ago? Let's see. I just looked it up. TWiV 200.
TWiV 200.
That was September 23rd, 2012. It was
10 years ago. Wow.
Me, Alan, and Rich, which is appropriate on today's episode that we have you. And you're sitting in that big building that's in the grass with the fence around it, right?
Exactly. I'm on the 5th floor.
Yeah. And of course, you were not there at the time, 2012, but we had
and importantly for new listeners, this is the home of a high containment laboratory, a BSL-4 laboratory, which will be, a feature of today's episode.
BSL-4, BSL-3, and BSL-2, right? Everything together, Mohsan?
Exactly. Yes.
And once again, since we're, what are we trying to do here?
We're explaining everything.
Trying to explain everything. Right.
These are containment levels for working with pathogens in laboratories.
BSL is biosafety level. BSL-1, you can eat it. BSL-4, you wear a spacesuit.
Yeah.
2 and 3 are somewhere in between.
Most labs operate at BSL-2.
BSL-1 is like high school, right?
Yeah.
So, we had a great time. We got to suit up. We got to work in the practice lab there. Oh, it was great. And then we came back a number of years later, Alan, for the opening, the official opening, right? And we did another podcast with David Quammen, I think, right?
Yeah, that was a good one.
Very good.
Yeah, I think that must be in 2018, I guess.
Could be.
Yeah, I think that's right. Because when we did the 2012 episode, the lab was not open. We were able to just walk through everything, and that was awesome.
Yeah.
But then 2018, of course, everything was airlocked in, and they were.
But I think you did the recording in the simulation lab, right? In 2012.
Yes.
Correct.
Right? Yeah, that's on the first floor. Yeah.
Simulation lab. Yeah, that's cool. It looks just like a regular lab. That's where we had put suits on and we walked in there through the door and so forth. That was a really great visit. At the time, our host was Ron Corley and Paul Dupre and Elke Mühlberger. had joined us on that TWiV as well. She's the only one left now of those three at the Needle, right?
Yeah, Ron Colley stepped down as
He
the
stepped down,
director.
right?
We have a new director now. Her name is Nancy Sullivan. She just started in December last year.
And Paul Dupree left.
Yeah, Paul Dupree left before I came here.
And Elke was on more recently to talk about low-view virus with a couple of other people. That was fun. So we have a particular affinity for the needle, I guess you could say. But today we're going to talk about your work, Mohsan, but I want to start by having you tell us all about your history, like where you're from and your training, all the way as far back as you can remember.
All right, Vincent, before I say anything about my history, I just want to tell you that I have been an avid listener of TWiV for the past 15 years.
Good.
I think I started listening in 2008 when I was doing my PhD. If I remember, it was only you and Dixon Depaumier. You talked about dengue virus. I think it was the 4th or 5th episode.
That's right.
And I have
Very
been hooked ever since. So
Cool.
I thought I will just start by saying thank you and thank to the whole team for all you have been doing for the virology community in particular and for science in general. So thank you for all that.
Yeah, you're welcome. I thought you were going to say something like, "I'm sorry, I can't talk. I have to leave."
This is how I'm feeling right now. But yeah, so I'm originally from Pakistan. That is where I spent the first 25 years of my life. I was born and raised in a small town called Shahkot, which is in the province of Punjab, and it has a population of around 200,000 to 250,000 people. Now, when I was growing up, the town was really famous for its high schools. So I really benefited from the quality and reputation of those schools and passed my matriculation with very good grades. Then I got admission in the college, once again in the town of Shahkot. But once I graduated from the college, then I moved to a bigger city to enter a university. And the university I entered is called University of Agriculture, Faisalabad. Faisalabad is the name of the city. And as the name implies, University of Agriculture has a lot of departments centered around the field of agriculture. I did my DVM from there, Doctor of Veterinary Medicine. It was a 4-year degree program, so I completed it and then did a job for 1 year just to get an idea of what was going on in the field. I worked as an assistant manager at a poultry farm, where my responsibility was to make sure that the chicken were vaccinated on time and they were properly taken care of. But I didn't really enjoy it. I think I didn't find it intellectually satisfying. So I decided to resume my education and move to the capital city of Islamabad, where I got admission at the Quaid-e-Azam University, which is the best university in my country, at least in the public sector. And there I did a master's in microbiology. And that's where I made a switch from animals to humans and did my research on non-polio enteroviruses. So the title of my thesis was The Prevalence of Non-Polio Enteroviruses in Cases of Acute Flaccid Paralysis in Pakistan. And I think the motivation behind this study was to find out which non-polio viruses will continue to circulate in the country after polio virus has been eradicated. So I completed my MPhil or master's. We call it MPhil instead of master's. And then I moved to Japan to do my PhD at the University of Tokyo. Although I was enrolled at the University of Tokyo, but I did my research at the National Institute of Infectious Disease. In Japanese, they call it Kokuritsu Kansensho Kenkyujo, which is a Japanese equivalent for the National Institutes of Health. There at NIID, National Institute of Infectious Disease, I joined the laboratory of Dr. Takaji Wakita, who had risen to fame because of his discovery of a hepatitis C virus isolate called JFH1, or Japanese fulminant hepatitis 1, that became the first ever natural HCV isolate. And actually, it remains the only natural HCV isolate to date that has the ability to grow in cell culture to very high levels. without the requirement for cell culture adaptive mutations.
Right.
His paper came out in 2005, and early 2007, I joined his lab. So there was a lot of curiosity about GFH1. And one of the fundamental questions was that what makes GFH1 so special, right? Why is it possible that the virus is inherently very aggressive? That's why it caused fulminant hepatitis, which is a very aggressive form of hepatitis where you see extensive liver damage within a few weeks after the onset of infection, often leading to hepatic coma or hepatic encephalopathy. So the question was that, is GFH1 inherently aggressive to cause fulminant hepatitis, and that is the reason it grew in cell culture, or is it that the virus did not cause fulminant hepatitis, but there was something wrong with the patient? There could be some genetic deficiencies or some comorbidities that led to fulminant hepatitis, and the fact that virus was able to grow in the cell culture was just a coincidence. So one of the senior scientists in the lab took on this question. His name is Takanobu Kato, and he did this research here at the National Institutes of Health in Bethesda, where he injected GFH1 into 2 chimpanzees. Okay, so one of the 2 chimpanzees received the patient serum from which GFH1 was originally isolated, and the other chimpanzee received the cell culture generated recombinant GFH1. Right. So the hypothesis was that if GFH1 if it was inherently aggressive, then both of those chimpanzees should get acute hepatitis accompanied by liver damage. But this is not what he saw. Both chimpanzees cleared the infection. Neither of them got hepatitis. But the virus persisted for quite some time. Like the animal that received the patient serum had viremia for around 9 months, but the animal that received the recombinant virus had viremia for only around 9 weeks. So I did my PhD thesis on in vitro characterization of virus isolated from these animals at different times post-infection. And one of the things I found was that the virus that caused persistent infection was not the original GFH1 that was being injected into these chimpanzees. The virus accumulated a lot of mutations early on, and those mutants were the ones that then led to persistent infection. I also found out...
And that was, which starting material was that?
So the starting material for one animal was the patient serum from which...
Yeah, but the one that caused the persistent infection, was that the patient serum?
That was the patient serum, exactly. So the infection persisted for a little longer time, like for around 9 months, compared to the other animal where it persisted for only 8 or 9 weeks. So that makes sense because the complexity, genetic complexity of the inoculum could be higher in case of patient serum compared to the cell culture generated GFH1. I think they use the word monotypic in the manuscript, but I think it is not monotypic. There are going to be quasi-species, but probably the complexity is going to be low compared to the patient serum. So I just did the in vitro characterization. I just looked at the replication kinetics of those mutants isolated from these animals and analyzed their ability to evade host defense mechanisms. So after I completed my PhD, I then came to the US to do my postdoc with Charlie Rice at the Rockefeller University. There I continued to work on hepatitis C virus. And one of the questions that I got interested in, and that is the question that has that intrigued and fascinated a lot of HCV virologists over many years, was the inability of natural HCV isolates to grow in cell culture. Right? As I mentioned, the GFH1 was the only isolate until then that could grow without cell culture adaptation. But the question was that what is this case with other natural HCV isolates? So when I joined the lab, Charlie had a hypothesis that might be the hepatoma cell lines that we use to grow hepatitis C virus express some restriction factors that do not allow HCV to grow in cells, and the cell culture adaptive mutations somehow help HCV to overcome those barriers. So I took on this project and set up an shRNA screen It was a pooled genome-wide shRNA screen, and the idea was to knock down those factors, whatever those factors were, and see if the cells become susceptible and permissive to infection. I spent quite a lot of time. Unfortunately, nothing came out of this exercise. But then I wanted to test the flip side of this hypothesis. I thought that it is quite possible that there are some essential host factors that are missing in these hepatoma cells, and they are absolutely essential for viral RNA replication. So then I set up a cDNA screen, an overexpression screen, and here I was able to identify a protein called Sec14L2, which when I overexpressed in cells, they became susceptible to hepatitis C virus. I think this paper was being discussed on TWiV when it came out in 2015, if I remember correctly. So this paper was published in Nature in 2015. And that was the time when I thought I was ready to move on to the next stage of my career. But that was also the time when a lot of therapeutic regimens were approved for the treatment of hepatitis C virus with the cure rate of around 95 to 99%. So some of my senior colleagues in the lab and in the field thought that because I did my PhD and postdoc only with hepatitis C virus, so it might be challenging to establish a lab around hepatitis C virus because there was some concern that the funding may go down and it might become difficult to secure funds to work on hepatitis C virus.
Don't be like Vincent and work on something that's being wiped out. Yeah, that's what they were telling you.
Yeah.
Or Rich, but yeah.
I think.
Yeah, the day I arrived and set up my laboratory, they declared smallpox eradicated.
Yeah, so I think World Health Organization has a hepatitis C virus elimination program. I think the target is 2030, which I think is a little too ambitious, but at least they have a.
The World Health Organization has a long history of ambitious targets for eradicating diseases. Yes.
Right. Anyway, so
Mohsan, I wanted to ask you, the J. What was the name of the isolate? JF1.
JFH1.
Is that the one where you made the infectious clone? The first infectious clone was made from?
Exactly. I was not the one who made it. Takaji Wakita made it. That paper came out in 2005, but I started my PhD in 2007, early 2007. But exactly, that was the first infectious clone.
That was a big deal because nobody had been able to do it before then. It turned out this isolate was the key.
Exactly.
Right?
Exactly.
Okay.
But then,
so as long as we're digging into this, okay, from what you've said, the implication is that the hepatoma cell lines used in Charlie's lab are different than the cell lines that were used in Japan.
No, the cell lines,
No?
the cell lines that were being used in Japan are the same. There was something special about JFH1 that it was able to grow in those cells. And JFH1 is the only isolate that was actually able to grow in those cells, right? But any other natural HCV isolate failed to infect those cells.
Because
so it really is the virus, not the cells.
It is the virus. It is the virus. Exactly.
Okay.
Yeah.
Okay.
But then, Vincent, many other people made derivatives of JFH1 where they took the non-structural proteins of JFH1 and appended them with structural proteins from either genotype 1, 2, 3, 4, 5, or 6. And they were able to make these sort of chimeric viruses, which have been heavily used in the field. So it seems there is something special about the replicase machinery of JFH1 that allows virus replication.
Okay, so it's just the replicase. The rest of the genome doesn't matter.
The rest of the genome doesn't matter much.
Okay.
Although
Got it.
there might be need for some compensatory mutations when you append the replicase region of JFH1 with other genotypes, but the difference is made by the replicase machinery.
Got it. Okay. So you stayed working on Hep C, or you changed? You changed to something else in Charlie's lab. I don't
So then I stayed back in the lab for another 2 to 3 years and developed an independent research program. And this program was focused on viral proteases. the idea behind this whole project was to identify cellular proteins that are being cleaved by viral proteases in virus-infected cells. Because, there are a handful of proteins that are already known as host targets of viral proteases, like Regai, MAVs, some other proteins, like even in some cases MDA5, TRIF, for hepatitis C virus. But there was no system described for global identification of these proteins. So I developed a platform that I call viral diagonomics, which is basically modification or adaptation of a protocol that was originally developed by Jim Wells' lab at UCSF to study apoptosis, which is a cellular process that is executed by a cascade of proteases called caspases. So I just adapted it and validated it for the study of viral proteases in the context of viral infection. I applied this method to cells infected with different enteroviruses like poliovirus, Coxsackie virus, EV70 that I obtained from your lab, Vincent, when I was back in New York, and then some human rhinoviruses. This led to identification of a lot of new proteins that were not previously known to be cleaved by enteroviral proteases. So when I came to Boston University, my original research program was to follow up on those proteins and then extend this approach to other positive strand RNA viruses like flaviviruses and alpha viruses.
Right. So that was, you came to Boston University in 2018?
I came here in 2019, early 2019. It was March
2019.
2019 when I joined.
And you brought with you the enterovirus project.
Exactly.
Right?
I brought with me enteroviruses and some other viruses as well.
That something happened in 2020, right? That kind of
exactly.
changed things.
Exactly. So I started here in March 2019. It took me around 5 or 6 months before we were able to start our experiments, like signing MTAs, bringing reagents, buying all the equipment that I needed, hiring people. I hired a lab technician and then a postdoc. And I think it was August or September when we did our first experiments in the lab. And then it took another 5 to 6 months. I think it was early 2020 when our research program started getting off the ground when the pandemic came along. And then in March 2020, when the COVID-19 was declared a pandemic, a global pandemic, Boston University announced to shut down its activities, including the research laboratories, with the exception of those that were working on SARS-CoV-2. So that led me to decide to pivot our research to SARS-CoV-2.
Like every other virologist.
Clearly
yes.
I didn't want to shut down the lab.
Yeah,
exactly.
Also, I mean, not only did you not want to shut down the lab, but how many opportunities are you going to get to study a pandemic virus that just emerged?
Exactly. And particularly when this building where I am is built for these kind of opportunities and these kind of situations, right? So that was a blessing because I was here having BSL-3 facility, BSL-4 facility. There were quite a few people who wanted to work on SARS-CoV-2, but they did not have those facilities. They did not have those resources.
Yeah. Sure.
So I think this really helped us because when I came here, BSL-3 was not really optimally functional. So I told myself that, okay, I'm going to work in BSL-2 initially to follow up on enteroviruses and slowly I'll transition to BSL-3. But then we had to do everything in a rush. It took us around 2 or 3 months, but then eventually we were able to begin our BSL-3 program.
So SARS-CoV-2 work is BSL-3?
It is BSL-3.
Okay.
So the paper I wanted to talk about with you today is Nature paper. Spike and NSP6 are key determinants of SARS-CoV-2 Omicron BA.1 attenuation. So everyone knows what spike is. NSP6 is one of the non-structural proteins, right?
Exactly.
So we talk a lot about Omicron and attenuation, and this idea that somehow it's remember at the beginning they said, "Oh, Omicron is mild. Go ahead, get it." But people dying are still dying of Omicron. So I'm wondering, what are your thoughts? I mean, do you think the human data clearly show that it's less virulent than previous isolates?
Well,
Kind of a leading question, I guess.
no. Yeah, I think there is some evidence, right? Let's first say that Omicron at the population level causes milder disease. I'm not going to say it's mild disease, but it's comparatively milder disease compared to the previous variants of concern. Now, there are two components of this observation. One is that Omicron has the ability to evade humoral responses elicited by previous infections or vaccines, right? But still, despite the fact that Omicron is able to overcome those defenses, but still, in the presence of those defenses, it cannot replicate to as high levels, I guess, as it would replicate in the absence of those responses, right? But then, the data in cell culture, in animal models, clearly indicates that Omicron, at least the BA.1 lineage, was attenuated. We have tested Omicron in many cell types in my lab. Other people have tested in their labs as well. We also put this virus in mice, in K18 human AS2 expressing mice. Other people have done the exact same experiment in other animal models, like in Syrian hamsters. And they have consistently found the same thing, that you get less severe pulmonary disease when you infect these animals with Omicron compared to either the Washington isolate, which is the prototype virus, the first virus isolate identified in the US in January 2020, or if you compare it with other variants of concern, including alpha, beta, gamma, and delta. So I think there are both factors contributing to the milder disease that we see at the population level. But I know that Daniel Griffiths, he does not agree that Omicron is any milder. But then there are other people. I remember there was an infectious disease physician, Vincent, who came on TWiV. I think his name was Jake Scott, right?
Jake Scott.
Jake Scott,
he had the other opinion, right? So I think
Daniel was not happy about that.
All right. Okay.
Well, there is, but there's also epidemiologically, there's a pretty strong line of evidence from the first Omicron wave, when there was this wave of infection in pretty much every country, that by every measure anybody's been able to do, whether it's test results or sewage testing, which I think is a better way to get these kinds of data, the scale of that dwarfed all previous waves.
Sure.
Just it was everywhere,
Sure.
and yet we did not have the ICUs overflowing.
Correct.
Yes, but remember,
Yes,
population
pre-existing
immunity.
right.
Population immunity was much greater, and the people getting infected were much younger because a lot of the older people had died. So
Had died, yes.
that had to be.
So
I remember the South African.
I agree. There were clearly multiple factors, but I think when you look at that and the data from animal models, cell culture, other analyses, and you put it all together, and organoids, and I mean all the systems people look at, there is something different about this virus.
Right.
I agree, it's something different. Yeah, I'm just not sure in people. But Mohsan, up till a few weeks ago, we were 500 people a day were dying in the U.S. from some of the later Omicron lineages. Is there evidence that some of the virulence has returned, do you think?
This is also, I think it's not really clear at this point, because I see papers going in both directions. Some people suggested when BA2, BA3, BA4, BA5 came along, some people suggested that they were more virulent compared to BA.1. But then there were other papers that were published in very high impact journals, which showed that they were very similar to BA.1 on both fronts, for their ability to evade humoral immunity as well as their ability to cause disease. So I'm not really sure if there are any differences, but there are suggestions on both sides.
Also, could it be at least a contributor that T-cell immunity, even though Omicron is evasive of antibody, maybe T-cell immunity is taking over, right?
Exactly. And I think there are papers.
Or non-neutralizing antibodies, FC-mediated functions, right?
Absolutely.
Okay.
And I think this is the idea, right? That the T-cell immunity might protect us for longer time, right? Because B-cell immunity,
sure.
at least when it comes to neutralizing antibodies, I think they protect us for around 6 to 8 months, depending upon who you ask. And then of course the memory B cell immunity that comes into picture. But people think that the T cell immunity is more long lasting, and I think people have seen T cell responses in patients who got infected with SARS-CoV-1, right? Many
17 years
after the original infection.
Well, I also think that the variants evade antibody immunity quite readily, but the T cell epitopes don't really change.
Yes.
So that's contributing as well.
Absolutely.
Okay. So this paper addresses this issue of, the virulence. And so why don't you walk us through what you did starting from the very beginning?
All right. So I can take you back to very early days of the pandemic and tell you how the SARS-CoV-2 research program evolved in my lab.
Sure.
And then we can come to this particular study.
Sure.
So I think it was January or February 2020. I received an email from a reporter from some local news channel telling me that they wanted to talk to me and get my opinion on what was going on in China and if we need to worry about anything. I said yes, without realizing what I was getting myself into, because I talked to that person and then I started getting a lot of emails, including some from CNN and like other media outlets. So just to be prepared.
Talking to reporters is a major cause of being called by reporters. Yes.
Yeah, that's what I learned. So in order to be prepared for those interviews, I started following the literature. I started listening to Tony Fauci, other infectious disease experts. And it was around the same time when I also started developing my own interest in SARS-CoV-2. And then I also started reaching out to people in the field, people who had been working with coronaviruses for a long time, just to get an idea of what kind of reagents are available and what kind of questions that should be addressed very early on. I mean, one of the people I would specifically mention, and her, Vincent, is Susan Baker, who is at Loyola University School of Medicine. And she has been working with coronaviruses for around 25, 30 years now. So I got in touch with her, and she has been really very instrumental in what we have been able to achieve over the past 3 years. So I reached out to some other people as well. I reached out to the director of Boston University's Center for Regenerative Medicine, Daryl Cotton, who is a big name in the differentiation of stem cells into lung organoids. So the first thing that came to my mind was to infect lung organoids with SARS-CoV-2 and see what kind of pathways are regulated upon infection. We also made a lot of human cell lines. Actually, the manuscript just got accepted last week. We had a panel of 18 cell lines that we engineered to express ACE2 and TMPRSS2 and analyzed their susceptibility and permissiveness to SARS-CoV-2 infection, identifying 1 or 2 cell lines that were really good, much better than Viro E6 cells that have historically been used in the field. Then we took 5 or 6 of those cell lines representing various body organs, infected them with SARS-CoV-2, and did global proteomic analysis to identify the processes and pathways that are responsive to infection. So one of the things that came out and that we followed up on was the interferon response pathway. We found out that 2 kinases, JAK and TYK2, which are associated with the cytoplasmic side of the interferon alpha receptor, were downregulated in SARS-CoV-2 infected cells. And when we did more mechanistic analysis, we found out that if not one, interferon alpha receptor 1 was also heavily downregulated in virus-infected cells. So what is the implication of this? If you infect cells with SARS-CoV-2 and let the infection establish itself, and then you expose those cells to interferon, you see little to no response. There is no induction of interferon stimulated genes, ISGs. But if you do the same experiment with uninfected cells, then you see a lot of response. There is induction of ISGs. So we propose that might be SARS-CoV-2 has the ability to continue to replicate in cells in the face of immune responses that are triggered in our body following infection. So these were the kind of studies that we were doing. But in late 2020, I think it was August or September of 2020, when Susan Baker wrote an R01 on SARS-CoV-2, and I was a co-investigator on that grant. Remember, by that time, there were no variants yet. There was no discussion about alpha, beta, gamma, delta, or Omicron. We just wanted to study some of the viral proteins for their ability to evade antiviral immune responses. So that proposal got funded in early 2021. In that proposal, we had suggested making various mutants. So the idea was to introduce functionally disabling mutations in some of the viral proteins and analyze the ability of those mutant viruses to replicate in cells and block antiviral innate immune responses. But when we started doing those experiments in 2021, I quickly realized that there was not an efficient reverse genetics platform. I'm sorry for using this word, Vincent. I know you don't like it. To make recombinant viruses, right? Now, throughout my career, I worked with hepatitis C virus, enteroviruses. I always used these kind of platforms to make recombinant viruses. I like to work with pure cultures, instead of patient sera, because you never know what is going to be in those clinical specimens. So I struggled a lot. We tried to implement various systems in the lab, but none of them worked.
Is this part of the reason for this, because coronavirus genome is so big?
Exactly. It is very big, and it is very unstable in bacteria. If you put it on a plasmid and then amplify in bacteria, you see a lot of deletions.
Okay. Been there, done that.
Yeah, of course. Yeah, with DNA viruses, right?
Cosmid clones of vaccinia DNA.
Right.
They just spit out big chunks of DNA habitually.
All right. Yeah. And then backmates as well, right? Is there something?
I've never worked with those, but my guess is I wouldn't be surprised for the same problem.
Right. So then a paper came out from a Japanese lab, Yoshiharu Matsuura's lab at Osaka University, where they described a platform called CPER, Circular Polymerase Extension Reaction. When I read that paper, I was really fascinated by the simplicity of the method. So I reached out to Dr. Matsuura because I knew him from my time in Japan, and requested all the plasmids and reagents that we needed to implement that system in the lab. So we received all those plasmids and started working on the system when Susan Baker connected me with Michaela Gack, who is at the Cleveland Clinic in Florida. She was also working on the CPER platform, and she was able to introduce some changes to the platform, some additional steps to CPER, improving its efficiency. So, of course, then we got in touch with Michaela Gack, and she was very kind enough to share her protocol with us. But when we applied that protocol in our lab, once again, we did not succeed. We couldn't get virus particles. At that point, we decided to screen our cell lines that I mentioned previously that we had generated in the lab and engineered to express AS2 and TMPRSS2. One of those cell lines changed the world for us because we were able to recover recombinant virus in 2 days. You do transfection of cells in 2 days, you get very high titer virus stock. And added advantage of that was that we didn't have to passage the virus, because for many other platforms, you rescue virus from the transfected cells, but the titer is so low that you have to passage the virus 2 or 3 times to get high titer virus stocks. But we didn't have to do that because we were already getting around 10 to the 7 plaque forming units per ml.
Just for our listeners, ACE2, of course, is the receptor for SARS-CoV-2, and TMPRSS2 is the plasma membrane protease that's needed for entry.
Exactly,
right?
exactly. I just call it an entry factor. TMPRSS2 is an entry factor, but ACE2 is the receptor that comes in contact with the spike protein.
Got it.
All right, so now by the time we had this platform ready, Omicron came along. This was in November 2021. Omicron came along, and immediately after Omicron was identified, two things became clear about this variant very quickly. One, its ability to evade humoral immunity. Second, its ability to cause a relatively mild disease in experimentally infected animals, and its poor replication in cultured cells. So I got excited. I remember in October 2021, in one of the lab meetings, I told my lab that, okay, let's cut down on our SARS-CoV-2 research and try to get back to our original research program, which really made them happy because we were having so many collaborations. And I have a small lab. I just started in 2019, and we were really burnt out in this process. But then when Omicron came along, I told them that, hey, this is something that I am really interested in. This is something that I have been doing throughout my career. And we have a very good platform to generate recombinant viruses. So why not to answer some important questions about Omicron? So one of the things that became clear was the spike protein was heavily mutated, right? If you align the genome of Omicron with the genome of the Washington isolate, you see around 50 mutations. Now, 60% of those mutations are in the spike protein. Spike accounts for only 12 to 13% of the total genome of the virus, but 60% of mutations were concentrated in the spike protein. So there was a lot of speculation that the spike protein is behind the attenuated phenotype of Omicron. And this is what...
But by the way, I want to interrupt you and ask you, where do you think Omicron came from?
Okay, this is a tricky question. I think one of the theories is that it came from an immunocompromised individual in South Africa. And another thought is that probably it was a reverse zoonotic jump. The virus went from human into minks and evolved in there, and then it jumped back into humans. We do not know the answer, and I think we will not know the answer.
Okay. Just wanted to see what you thought. Okay, go ahead.
So my initial thought was that, okay, spike is going to be responsible for the attenuation of Omicron, but let's test it experimentally, because at that moment it was just a speculation. But the important question in my mind was to find out what is the evolutionary trade-off between gaining the ability to evade antiviral innate immunity and the ability to replicate to high levels. In other words, I wanted to see if the mutations responsible for the ability of Omicron to evade innate immunity were the same mutations that were responsible for the inability or lower ability of Omicron to get into cells.
Right.
But for that, we first have to make sure that spike was behind the attenuated pathogenicity of Omicron. So the first experiment we did that got us into trouble, unfortunately, was to take the spike of Omicron and put it into the Washington isolate backbone. But I just want to point out one thing before I get into the experimental details, that before we did this experiment, we got all the approvals. I wrote a very detailed protocol outlining very clearly what kind of experiment we wanted to do and why it was important to do this experiment, and sent that protocol to the Institutional Biosafety Committee of Boston University. They approved it. They also sent it to Boston University Dirk Committee. They also approved it. And then
dual use research of concern.
Dual use research of concern. They also approved it. And then in Boston, you have another layer of regulation. Once your Institutional Biosafety Committee approves a protocol, it is being sent to Boston Public Health Commission, BPHC, and they also have to approve it before you can begin the experiment.
Here in New England, we love our layers of government.
Exactly. So
But Mohsan, should it have been approved by NIH as well?
so this is something confusing, Vincent, because the research, the animal experiments that we did, the cell culture experiments that we did, they were all done with my startup money, because I still
see.
have my startup money. I did not have money to work on Omicron.
Got it.
So at that time, we didn't feel the need.
I would have thought that would be, maybe I misunderstand this, but I would have thought that would be part of the responsibility of the Institutional Biosafety Committee to look at something like this and say it ought to be passed on, kicked up to NIH.
No, only if you have federal funds. Only if you're using federal funds.
But if you do have federal funds, right. This is a special case.
But he didn't, so.
But just to clarify, if you do have federal funds,
Exactly.
if the IBC looks at this and says, we're not sure, do they have a responsibility to kick it upstairs to NIH and have it looked at?
Yeah.
I think this is a pipeline. If you tell them,
I believe that's correct. And I would add, my understanding of this process is NIH has effectively delegated its authority to the institutions on this because they don't have the staff to review every single experiment in every single grantee's lab. And so this would only get sent up to the NIH if your institutional board, which is vetted by the NIH on a regular basis, and they have these discussions. If they looked at it and said, maybe this is a little on the bubble, then they can send it up to NIH.
Or if they said, this is Dirk, in our opinion, they would send it to NIH.
They would send it, right.
Exactly.
Because I'm on the Columbia IBC, and that's how we work. If we think it's fine, then it stays there.
Yeah.
Right.
Yeah, I don't think the review would have been any different if you had been on an R01 with this project. That's just my speculation from interacting with the regulators.
Exactly. So when I submitted my IBC protocol, I told them what money I'm going to use for this. So I said we are going to use our startup funds. So I think that is why they didn't feel the need to go to NIH.
Mohsan, let me tell you, these are the nuances that the press often don't get, right?
Oh, yeah.
Right. Exactly.
Yeah. I mean, unfortunately, gain of function itself has become a dog whistle that only a minor percentage of individuals understand what it actually means or what the subtleties are.
Right. But I would actually argue that even if you had been doing this on an NIH grant, I suspect your institution would not have kicked this up to NIH because, I mean, looking at this as a virologist, this is the obvious experiment to do. And from what you've told us about your lab's capabilities, your lab's a pretty obvious place to do it.
Yeah, for sure.
So this all fits.
Right. I mean, this is, as I said before, this is one of the purpose for which Needle was being built, just to make sure that we are able to respond to outbreaks and pandemics.
Were these experiments also all done in BSL-3?
All of these experiments were done in BSL-3.
Okay.
Right. Yeah. But what you said, Rich, I think this is really hurtful because when the pandemic came along, I was under the impression that people are going to value the importance of virology. I mean, I remember all those conversations with my friends. That was the first time my mom knew what virologists do. Okay, what a virus is. And I was so happy that finally I was able to communicate with my family about what exactly I do in the lab, right? But unfortunately, the things went in the other direction. And I think the word gain of function has been heavily mischaracterized.
Yes.
Sure. Totally agree.
Yeah. I mean, first of all, the term was borrowed from the field of genetics and applied on virology, where it is inexact, unsatisfying, and often misconceived as something sinister. Like people think that the gain of function experiments are meant to have some sinister designs, right?
Yeah.
But if you think about these experiments, I think they are at the heart of most of the research we do in virology, right? I mean, think about all the vaccines we have been able to make. Whenever you manipulate the virus to increase their yield or their immunogenicity, it is gain of function.
Sure.
Oncolytic viruses, right?
Sure.
So
So, Mohsan, here's a thought question. If you attenuate a virus for a vaccine, we would look at that as a loss of function, right?
Right.
But it is a different function.
Exactly.
So maybe it could be a gain of function too.
Absolutely.
That's what you mean by being kind of a hazy thing, right?
Yeah. If it is becoming more immunogenic in that process, it is gain of some sort of function, right?
Yeah. I mean, your experiments that you were going to do with the spike, you had no idea if they would be gain or loss,
Exactly.
right?
And this is an important point, Vincent, because in the IBC protocol, we said that when we infect our cells with the chimeric virus, if we see any evidence of increased replication compared to the backbone virus, we will immediately stop our experiments and we will report to IBC. Okay, and we will also report to BPHC. Right? So this is a language very clearly written in my protocol, and I think this is the requirement. This is what you can do, right? As you said, you don't know beforehand if the virus you are going to make is going to have a gain of function or loss of function. But if you see any evidence of gain of function, then you have to stop that experiment. But there is a complication in here. I can give you an example from my own lab. Okay. So we made this virus. It has an Omicron spike, but all other proteins from the Washington isolate. Now you infect lung cells with this virus and compare it with the Washington isolate. We found out that this virus was attenuated compared to the Washington isolate. It replicated poorly compared to the Washington isolate. So we didn't feel the need to report it because it didn't cause a stronger infection compared to the Washington isolate. But now, listen to this. When we did the exact same experiment in the upper airway epithelial cells, the phenotype was opposite. The Washington isolate replicated poorly, but the chimeric virus with the Omicron spike replicated way better than the Washington isolate. So now what to do here, right? It is cell type dependent. In one cell type, it is replicating poorly compared to the backbone virus. In the other cell type, it is replicating better than the backbone virus. So that's why I just feel that, this definition of gain of function, I think it does not really apply very well to virology experiments. And we have to come up with a better definition or better way to do these experiments or regulate these experiments.
And I think, if I'm not mistaken, you obviously know much more than I do about this, that NIH has a very narrow definition of gain of function with respect to regulation of experiments, whereas the public or the congressional understanding of gain of function is not nearly so narrow. I'm thinking of this now famous argument between Ron Paul and Tony Fauci, where Ron Paul's holding up this paper saying this is gain of function, and Tony's saying no, it's not. And you can kind of understand how somebody who didn't understand the subtleties would look at this and say this is gain of function. But Tony's saying no, NIH has a very strict, well-defined definition of gain of function because it needs it in order to filter what are appropriate experiments and not, and this does not fit that definition.
Exactly.
So it's really, for the public, extraordinarily confusing.
It is because,
and I appreciate how frustrating it is for you. I appreciate that story.
It is frustrating because I worked on hepatitis C virus for most of my career, and I know some groups when protease inhibitors were developed as potential anti-HCV antivirals, people grew hepatitis C virus in the presence of those protease inhibitors in order to search for escape mutants, right? And once they identified those escape mutants, they then optimized the drugs. They then improved those drugs in order to make sure that they are able to work against those escape mutants as well. I know people who want to do the exact same kind of things with SARS-CoV-2, but they are scared. They do not want to do those experiments only because they know that there could be a backlash, right? And this is not good for science.
That's a great example because those hepatitis C drugs are absolutely critical for public health,
Exactly.
right? And those experiments were critical in improving those drugs. Wow,
And
Well,
great example.
Mohsan, someone at Columbia wanted to do those SARS-CoV-2 experiments, and the experiments came to the IBC, and we said these need to be done. So we allowed them to go forward because fortunately we had some virologists on the committee, and we recognized, as you said, the importance of doing that.
Yeah, because the virus is going to do that experiment for you.
It will.
And you'd rather have it done in the lab where you can see it and autoclave it afterward and understand how to respond to it before it shows up in a patient.
Absolutely. Yeah, so I don't know where I was with the.
Well, so we can go back to the paper actually, because you're talking about, so you put spike into
so
the Washington isolate.
exactly. So we can go
You
back
saw
to the
these
paper.
differences.
Yeah, so we made this
Yeah.
virus. We got all the approvals. We made this virus. We tested it in 2 different cell types initially, and we found out that the chimeric virus was it didn't replicate to as high levels as the Washington isolate, but it also didn't replicate as poorly as Omicron.
Okay, somewhere in
So
between.
it
right?
gave us an intermediate phenotype. Another interesting observation we made was that the plaque size was different. The Washington isolate made large plaques. Omicron made pinpoint plaques, but then the chimeric virus made intermediate-sized plaques. Okay, and this was reproducible whenever we did this experiment, because whenever we made a virus, we had to do the plaque assay, and we saw this phenotype consistently.
Mohsan, this is great because we see so few plaque assays in SARS-CoV-2 research. And there's, as you say, they really tell you a lot.
Exactly.
And I'm so glad to see this.
Yeah, no, it's one of my favorite assays, So then we also competed them against each other. So we made a recombinant Omicron with one fluorescent protein and our chimeric virus with another fluorescent protein, infected cells with both viruses at a 1-to-1 ratio, and then looked at one color and the other color, like how the percentage of one color compares to the
See
other
who
color.
wins. Yeah. Who wins in a fight?
Who wins in
Yeah.
a fight? So you basically pit 2 viruses against each other and see who wins. And our chimeric virus came out as a clear winner compared to Omicron, once again telling us that spike was not the whole story. The spike was not the only protein behind the attenuated phenotype of Omicron. So then we did our in vivo experiments. They were done in collaboration with a friend of mine and a very smart virologist here at the Needle. His name is Florian Duam. He works with a lot of animal models. So we worked with his lab and tested our viruses in K18 human ACE2 expressing mice. Now, when we infected these animals intranasally with the Washington isolate at the dose that we used, all animals perished. The survival rate was 0%. But when we infected them with Omicron, the survival was 100%. But when we infected them with our chimeric virus, then the survival was 80%, right? So that means that even this virus was able to kill a lot of animals, once again suggesting that spike was not the sole determining factor behind Omicron's attenuated virulence. So another observation we made while doing these experiments was pretty interesting. Washington isolate mainly infected the alveoli in our lungs, okay? Whereas the chimeric virus that contained the spike of Omicron, it mainly infected the bronchioles, right? And it might have something to do with the lack of Omicron spike, lack of ability of Omicron spike to use TMPRSS2 mediated route of entry, because TMPRSS2 route of entry is the prevalent route of entry in alveoli, whereas in bronchioles, I guess it is not published. But from my discussion with some of the colleagues in the field, they think that the endosomal pathway is more active in bronchioles. And there are studies showing that Omicron is deficient in utilizing DMPRSS2 mediated pathway. So that might explain the phenotype that we saw. But overall, from all these in vitro and in vivo experiments, we drew a conclusion that spike was not the only factor behind Omicron's attenuation. So this was the study that we submitted to Nature in March 2022. Then it went into review, and then it went into revision. But one of the things the reviewer said was that, okay, it's a negative data because it clearly tells that spike is not responsible for Omicron's attenuation. If it is not spike, then you have to find out what else it is. And luckily, by that time, we were already doing these experiments because that was the logical extension of our study. By the time we had reviewers' comments back, we had already figured out that it was NSP6, right? So we did in vivo experiments. We spent 3 months working on the reviewers' comments and generating these additional data to show NSP6 was the actual determinant along with spike. And then we submitted the revised version. It went into review. It went back into review, but then it took forever. I remember we submitted this manuscript in July. We didn't hear back in August. We didn't hear back in September. And then October 13th was the day, which I'll come back to because that is when the whole controversy happened. So what we did to identify NSP6 as a critical factor, and I think this made a difference. We took our chimeric virus and used it as a backbone to begin to combine Omicron spike with one Omicron non-spike protein at a time, right? So the spike was always there. And it was there because our in vitro experiments showed us that if you have spike of Omicron, you already see attenuation, quite significant attenuation, right? So we just wanted to see which other protein could bring the virus down to the level of Omicron. So we combined with NSP3, NSP4, NSP5. We didn't see any difference or very little difference. But when we combined spike with NSP6, then we were able to get the virus that had very similar replication kinetics as Omicron. Then we did in vivo experiment, and there we saw around, I think it was like around between 70 and 75% survival with this chimeric virus, leading us to conclude that spike and NSP6 work together to yield the phenotype that we were observing.
So those were loss of function experiments.
These were loss of function experiments. They turned out to be loss of function experiments because one of the reporters at one point sent me an email saying that Boston University is saying that these were loss of function experiments, but were you really disappointed when they turned out to be loss of function experiments because your intention was to have gain of function, but when there was loss of function, were you really disappointed? I didn't reply to that email, but
no, your goal was to find out what loses
the rest
function.
of the loss of function.
My goal was to answer a biological question, and as I pointed out in my IBC protocol, if I had seen any gain of function, I will not proceed. I will just stop and let the IBC know.
Alan, what do you think of that kind of a question from a reporter?
Yeah, so that's a question that stems from a fundamental misunderstanding of what the experiment was. And I'm guessing that was a reporter who didn't have a background in this, who probably got pulled in to cover the pandemic and was doing what they could, having been pulled off the sports desk or whatever their normal beat is.
So once again, in the interests of explaining everything, NSP is non-structural protein. Viruses have a lot of genes that do stuff. Only a few of those genes wind up in the mature virus particle. Those that don't, in particular, if you don't know what else they might do, get called non-structural proteins and given a number. So they're made during the infection. They do something. We don't know what it is. Tell me about NSP6. What does it look like? What do you think it does? Because this is interesting. It's part of the attenuated phenotype.
Yeah, SARS-CoV-2 has 16 non-structural proteins.
16.
Exactly.
Holy
1
cow.
through 16. And many of them are made as a polyprotein. There are two polyproteins. One is NSP1 through NSP10. And then there is a ribosomal frame shift where we get another polyprotein. And that polyprotein has NSP1 through NSP16, right?
So by polyprotein, you mean that it's initially made as one big long thing.
Exactly. And then
cleaved?
And then it is chopped into pieces by two viral proteases. One of them is called papain-like protease. The other one is called main protease, liberating these individual proteins, 16 individual proteins. Now, NSP6 was a biggest surprise. I would not have been able to predict that NSP6 will be the culprit. I was mainly thinking about some of the structural proteins, mainly nucleocapsid, because that had quite a few mutations. Now, NSP6 is a transmembrane protein. It has 6 transmembrane domains.
How big is it?
It is around, I think, close to 300 amino acids.
Okay, it's big.
Yeah, it is. I don't remember the exact number.
That's something like 100 kilodaltons or something.
You might get emails or letters, but I think it's in that range. It is close to 300. The N-terminal and C-terminal ends are on the cytoplasmic side. And then, of course, there are loops. Some of them are on the cytoplasmic side. Others are on the ER luminal side. Now, there are only 3 non-structural proteins that have transmembrane domains. NSP3, NSP4, and NSP6. All other non-structural proteins are soluble. Okay, and NSP6 has been implicated in the formation of replication organelles. Those double membrane vesicles where HCV RNA replication happens. Actually, there was a paper. It came out in Nature in 2021, I guess, where the authors showed that NSP6 is responsible for anchoring those DMVs on endoplasmic reticulum. So it seems to have a zipper-like activity. Of course, it oligomerizes and makes sort of zippers that connect DMVs with endoplasmic reticulum. And the authors in that paper suggested that these zippers can serve as channels for a food supply, for supply of certain things from ER to these DMVs. The surprising thing is that so there are 2 changes in NSP6 of Omicron compared to NSP6 of the Washington isolate. One is a 3 amino acid deletion, which is on one of the loops on the ER luminal side. Then there is a substitution. I think it is isoleucine to valine that is on another loop, but once again on the endoplasmic reticulum luminal side, right? So at this point, I have no idea what exactly these mutations do. We are working on them in the lab. But I would like to point out that these deletions of 3 amino acids, they have been seen in many other variants of concern. Okay? So it is a deletion of 105 through 107th amino acid, like 105, 106, and 107. These 3 amino acids are deleted in many variants. The only difference in BA.1 is that the deletion is shifted by 1 amino acid. So it is 106 through 108 instead of 105 through 107. But then it went back to 105 through 107 in BA2 and subsequent variants of concern. So we really do not know why this shift happened. Actually, Susan Baker and my lab, we recently wrote a short summary on the evolution of NSP6. We found out that in SARS-CoV-like viruses that have been isolated from bats and many other species, there is no deletion in NSP6. But in humans, most of the SARS-CoV-2 isolates, I think with the exception of one variant, if I remember correctly, there are deletions, the same deletion, the same region. So we think that this deletion might be responsible for or might be one of the factors for adaptation of SARS-CoV-2 to the human population. But at this point, we do not really exactly know what these mutations do.
How about the substitution? Are you trying to sort out the relative contributions of those mutations?
So those are the experiments we are doing in the lab right now.
Okay.
But one thing that happened just a few weeks ago, or might be a month and a half ago now, Melanie Art and Jennifer Duartna, they published, they put a preprint on bioRxiv where they did the exact same experiments that we were doing. They used a different experimental model, and they also showed that NSP6 was behind the attenuation of BA.1. So this was very satisfying and very gratifying for us because, honestly speaking, I was not as happy when our paper came out as I was on seeing the preprint because it really validated our findings.
Did they get in trouble like you did for doing these experiments?
So this is interesting. No,
Of course, she has a Nobel Prize, no problem. But who knows why?
Could this be related to the differences you see between bronchiolar and alveolar replication of this virus?
So that difference is being created by the spike.
That's all spike.
That is all
right?
spike.
Okay.
But exactly how and why Omicron spike has more predilection for bronchioles compared to alveoli, that is another thing we are trying to figure out in the lab.
But asking Alan's question another way, do you know sort of in terms of virus replication or, cell biology, why it is that this NSP6 mutation is making a difference?
Yeah, there's clearly some selective pressure to delete these amino acids when you're in humans,
exactly.
Right?
No, I do not know the answer. This is something we wrote in the discussion saying that it is quite possible that these mutations are some sort of adaptation for Omicron replication in upper respiratory tract. And NSP6 mutation is just a compensation for that attitude of Omicron, but we do not know for sure. And
this is just boggling the mind because, you're touching on how these viruses cause disease. What could be more important? And how the hell else are you going to do the experiments?
Exactly.
That's the central question of the pandemic. Yes.
Yeah.
Well, as Alan said, they probably came from the sports desk, and
a lot of folks got pulled in to cover this and didn't necessarily, and it also depends on who they talked to before. I mean, if they just got off the phone with one of the anti-gain of function brigade who was on about this, then that would be their fixation.
Oh, of course. Yeah. By the way, Mohsan, if you just take NSP6 from Omicron in the Washington background, does that have any different properties?
Yeah. So this is what is going on in the lab right now. But I can tell you that Melanie Art's preprint, they used replicons. Okay, to look at the NSP6 contribution. They did this experiment and they saw the difference. I do not remember if their backbone was Delta or the Washington isolate, but when they put NSP6 of Omicron, they saw attenuation, and there was no spike in that case because it was just a replicon, just non-structural proteins.
All right, I want to see in your hands because you're looking at the whole cycle. And
exactly.
I want to see effect on yields, and I presume you may put them in mice as well at some point.
Yeah, those experiments are in progress right now.
And plaque sizes.
And plaque sizes. So this is an important point, Alan, because when we combined the spike and NSP6 of Omicron, we got the same plaque size as Omicron, right? So it was not only the replication kinetics and in vivo phenotype, but the plaque size also was similar to what we have been seeing with Omicron.
Cool. So you said something happened on October 13. What happened?
Yeah. So so I just wanted to walk you through the timeline of how exactly this controversy happened, right?
Okay.
So, when you submit a SARS-CoV-2 related manuscript to Nature these days, if they decide to send it out for a peer review, they send you an email asking you to share the preprint of your paper on some public server of your choice. It could be bioRxiv, it could be Research Square, or any other server. They sent me an email in March, but I ignored it. Just because I have a mixed opinion about bioRxiv. And back in 2020, we didn't have a very pleasant experience when we posted our preprint on bioRxiv. So I ignored it. And then, on October 13th, I was in a seminar when I received an email from the editor. She said that, Mohsan, I'm just trying to complete the checklist for your manuscript. And in this process, I was trying to find out your preprint, but I didn't find it. Can you please send me the link of your preprint? So that was the time when I told her that I never posted it. And she was not very happy. She said, please do it as soon as possible because we have signed this mutual statement, which requires us to make sure that all of these papers are shared with the general public as soon as possible. So I remember I did it while sitting in the seminar. I I uploaded it, and then I received an email from bioRxiv saying that it's going to take around 48 hours before it goes online.
Mm-hmm.
I sent that email to the editor, and the very next day, it was Friday, October 14th, she sent me an email saying that thank you for sending that email. I just want to let you know that your paper has been accepted for publication in Nature, but I'm in a rush. I'm going to send you an official letter of acceptance on Monday. So of course, I was very happy. I shared it with my lab. I shared it with some of my collaborators as well. And we were over the moon. This was our first Nature paper from my lab. And then came Saturday. I went for a hike with some of my friends. And I was in the mountains when at some point I pulled out my iPhone from my pocket and opened the email. And I saw an email with a very sort of unpleasant language. It said something along the lines that you scientists have no regards for the general public, and you are bent upon wreaking havoc with the human health and all those sort of things. I could not make a connection. I talked to my friends and they said, oh, this is some sort of random misdirected email, just ignore it, which I did. And then on the way back, I once again checked my email and there were more emails. And they all have very decorated language. One of those emails mentioned our preprint. And that was the first time I realized that something is going on with the preprint. But once again, I was unable to understand what exactly happened. So when I came back home, I opened the bioRxiv website, and there was a lot of Twitter activity going on. In just like 2 to 3 hours after the paper came out or the preprint came out, there were already 500 to 600 tweets, which when I opened, they were really nasty, okay, very strong language. And that ruined the whole weekend because I also started getting emails, some threatening emails. And then on Monday, the things went to a whole different level because then this tabloid British newspaper called Daily Mail, they put together a piece with a very inflammatory language.
The Daily Fail, I've heard many Brits call it.
I think this is a better name. But they put this piece saying that at Boston University, scientists have made a virus that have a kill rate of around 80%. and that was when the whole hell broke loose. I started getting a lot of emails, and Boston University honestly came under assault because there were so many emails and very unpleasant emails, very aggressive emails, and I started getting a lot of threats. Okay. But I just want to give a shout out to the Boston University leadership administration because they very quickly looked into all the approvals we had obtained before we did this work. They also realized the importance of this work, and they learned that the paper has already been accepted for publication in Nature. They also knew the facility where we did all these experiments. So then they stood very strongly with us. They refuted the claims made by all these newspapers. They fielded queries from various news media channels. So they did all they had to do to make sure. I mean, not only Boston University administration, but Boston University security as well. They were also very vigilant and they made sure that people feel safe. This thing really continued for around, I think, 7 to 10 days before it subsided. And I think then they found someone else.
Some new target. Yes, that's the cycle. It lasts about a week, and then there's some new target.
But many people from the field reached out to me, some really big names, just to extend their support. Some of them took time to talk to me on phone or via Zoom to share their own stories. how they went through hell after they published papers. Honestly, I was very naive. I didn't realize. I knew something was going on out in the public domain, but I was not aware of how much it was and what kind of impact it was having on the scientific community. But I think it was a traumatic experience. It was an awful experience. But I think I got exposed to a whole new world of experience, and I think I learned certain things out of it.
Well, thank you for sharing this, because this is really an important story, in particular in context of this whole episode, where at the same time you can see the value of the experiments and what they actually were. I appreciate this. I'm sitting here thinking, oh my goodness, you're being attacked, Alan.
I'm sorry, I'm under attack by a wild animal. For those of you who are listening on audio,
I'm sitting here thinking that when people start talking to me about gain of function, I'm going to point them to this episode and say, listen to the whole thing. Okay, this is important.
So, Mohsan, what about the NIH? Some of these headlines said, NIH should have been consulted, blah, blah. So did you eventually sort that out?
Yeah, so that has been sorted out. The thing is that we cited NIH grant. It is the same grant that Susan Baker has, and I'm a co-investigator on that one. We thought that we used NIH money to put the system in place, because all the reverse genetics platform that I talked about, our struggles in 2021, with some other platforms, then with the CPR, we used NIH money to develop that platform. But then we used that platform for something other than that we had promised to use it for in our grant proposal. So we thought that we should acknowledge NIH for their money, at least, for the development of the reverse genetics platform. But then, of course, NIH has new guidelines. I think they came out in 2021. We were not aware of those guidelines, but NIH sent those guidelines to us. And based on those guidelines, we changed things. We removed that grant. And there was another grant from another collaborator. They also changed that grant. I mean, they did not make the virus. The virus was made in my lab. But NIH, so they also removed their grants.
So as far as NIH is concerned, you didn't do anything wrong.
Exactly.
Right?
Exactly.
Okay.
Yes.
And all the negative headlines all stem from a misunderstanding of the experiments, essentially. They don't know what you were doing. They don't realize that the virus you made is less pathogenic and less cytopathic than the Washington strain, right?
Exactly. And the interesting aspect of this whole thing is, Vincent, that we were not the first one to make this virus. This chimeric virus with the Omicron spike in the backbone of the Washington isolate was already published by the time we put our preprint online. And not by one lab, but by many labs. Right? So that's why this idea or thought never crossed my mind that we were going to be in trouble for making this virus.
It's always about being in a certain place at a certain time, right, Alan? With the way the press is viewing something, they talk to one person who sets them off, and that's it, right?
And I don't necessarily want to go here, and it's an uncomfortable topic probably, but have you considered that your national origin may have played into some of this discussion?
I absolutely think so.
Because it's a distinct possibility.
Yeah.
Which is really just
disturbing.
Kind of the state of toxicity that we have, especially in this country, but in many other countries as well right now.
So, Mohsan, if you were to do it again, what would you do differently?
So this is a good question, right? And this is something I have been thinking about as well. I asked this question to BPHC as well. I asked this question to our institutional biosafety committee as well. And my understanding is that whatever we did last time, we did everything that we were supposed to do last time when we did those experiments, right? If we have NIH money, then we should inform them. If we do not have NIH money, then we do not need to inform them. There were some people on internet who said that even if you don't need to inform them, you should inform them, right? So this is a debate. And I know that NSABB and many other forums, they are debating about this topic, and there is conversation about whether this kind of research should come under oversight, NIH oversight, only if NIH is supporting their research, or if even if they are not supporting their research, it should still come under that oversight. So like once the regulations and guidelines become clear, then of course we will follow it. But at this point, I think we are under no obligation to inform NIH if we are not using their money to do research. This is what my understanding is.
And it's not just a question of you don't want them to say no. I would argue that informing them of research that they are not funding is placing the NIH in a law enforcement role, which is not something they are constituted to do, and is not a precedent I would encourage researchers to start setting.
Right. Yes, so this is why, I mean, you must have seen NSABB guidelines. I think they were recently approved.
Well, they were suggested, but they haven't approved them because they have to be rewritten.
Yeah, I think conditionally approved or something. But the language there is vague, and I think that language has to be vague because NIH is also in a very weird position, right? They have on one hand they have to make sure that the science is not compromised, but on the other hand they also have to make sure that people feel comfortable, right? They know that the research is under regulation and oversight, right?
Sure.
Yeah,
well, they have to answer to Congress, unfortunately, so they have to really be very careful.
Well, no, I would say fortunately. I think it's appropriate that since the public is paying for the research, the public should have oversight of how that's being operated in a general sense.
If Congress were fair, Alan, I would agree.
Yeah, I mean, there are issues with the structure of our Congress that are never going to be fixed, but that's...
I think the only thing I want to say on this topic, the only last thing, I guess, is that it is okay. You know, if the scientific community feels the need for more regulations, go for it. But they should be based on a rational discourse. You know, they should not be done under some sort of pressure or fear, because at the end of the day, I think science is at the core of our civilization, right? I mean, I was just thinking about it, that during the first 2 decades of 21st century, we have seen at least seven viral outbreaks. We had SARS-CoV-1, then we had MERS-CoV, then we had SARS-CoV-2, then we had a Zika virus outbreak in 2015, 2016. Then we had a flu outbreak in 2009, and we have at least 2 outbreaks of Ebola virus. And actually, a Marburg virus outbreak is going on right now. And all of these viruses that I have enlisted, they came from animals, you know?
And that's just the ones that directly infect humans.
Exactly.
We also have an African swine fever virus pandemic that is decimating swine herds that we've talked about, and we have other viral issues that are clearly a problem.
Absolutely. So I think if we do not want to compromise the ability of the scientific community to deal with these future pandemics, then we need to apply our mind and think what is appropriate. I just feel that we need to further strengthen the scientific community. We need to equip them even better so that we can deal with these outbreaks in a better way.
So I totally agree with you, Mohsan. The problem with the NSABB is that it's often hijacked by a couple of individuals who have some agenda. They think no scientist can do any experiment safely, and we should severely restrict what we do. And I don't understand why a few people dominate the agenda. And that, I mean, we obviously can do things safely, and I agree we have to have a rational discourse, but these individuals don't have a rational discourse. They say you can't do experiments any longer, and that's poisoning the discourse, you know.
That's very true. Some people have loud voice. This is exactly what happens with the preprint as well, because when I looked at the first few tweets, those people had thousands of followers. Okay, I have only, I think, a few hundred followers. I am not be able to compete with them, right? And
No.
then it was a vicious cycle, someone tweeting your story with 20,000 followers, and then one of those followers
Yeah, of course.
retweeting it, and they
Yeah.
are having their own 10,000 followers. So it spread like fire.
No, it's very difficult. I mean, I have 160,000 followers, right? So I judiciously try to tweet things the last couple of weeks about how there's good evidence that SARS-CoV-2 came from nature, right? And then you look at the responses. There are people who are just saying, you don't know what you're talking about. You have no knowledge of virology, blah, blah. And then, so it's not the scientific forum that we are used to. Although at meetings, things can get heated as well, which is fine to discuss things. But this is crazy to make it personal, right?
So what this exactly tells us then? Like, do you think that we need to communicate more? Like there should be more communication between the scientific community and general public?
I think so.
Because
So I think I think you're right. I think that for the most part, scientists have been quite silent. We had this wonderful community, insular, among ourselves. We don't talk to people much, and people don't know what we're doing, because now that we and others are communicating, we get people telling us, for once I find out, what scientists are doing and what you're like. And it turns out you're just like us. So I think it's important for more of us to do that.
Yeah, because when I was getting all those emails, at least there were a few emails which made me feel that the senders were not trying to be aggressive, but they really wanted to understand why we did that research and what is the importance of these kind of experiments.
Well, that's why we wanted to have you here. And so Susan Baker actually strongly encouraged me, which I really am grateful for, because this has been a very illuminating discussion. The science is extremely interesting and important. And it's also has been important to get your experience out here. And, hopefully some of the people who wrote these headlines will listen and realize their error. So unlikely, but we hope.
No, they're already writing the next set of enraging headlines because rage sells. That's the unfortunate loop we're in now.
But I think, Mohsan, communicating at any level of every scientist has to communicate to some extent. You don't have to do a podcast. You don't have to do 8 podcasts, but you have to do something.
Exactly.
Otherwise, we're going to keep getting in this trouble. And the pandemic really amplified what a problem it is, right? Because people do not trust and they don't understand.
I think our language is also very different, right? These people who are anti-science or anti-vax, they speak with a lot of confidence and trust. Scientists, they use a lot of highly likely, maybe, probably kind of expressions.
Yes.
And when it comes to public speaking, I think this does not convey a good message.
No, it doesn't. Right. I try to tell people to back off that, because that's a big problem. But listen to this story. So the other night on the live stream, we were talking about mRNA vaccines, and someone came on and wrote, you guys don't know what you're talking about. These are horrible vaccines. Did you know they cause class switching? And in full seriousness, thinking that was a big issue, right? So this is the problem we have, right? Class. What do people know about class switching, right? The headline
and to be clear, class switching is something the immune system is supposed to do with antibodies as an immune response matures. And I think you can look back on past TWiVs, look at episodes that have Brianne on, and I think you'll find information.
I was kind of hoping that being vaccinated with an mRNA vaccine would change my socioeconomic status.
Yeah, good luck with that. Well, so I think that it's our mission here, Mohsan, is to try and do our little bit to communicate and maybe encourage other people to do it in our fields, right? Because you can only do it in your field. You don't know enough about other things. So,
exactly.
Anyway, Mohsan, Boston University, the Needle. Thank you so much for joining us. Great story.
Well, thank you for having me, really. It's much
great.
Appreciated.
And when you have new results, we'll have you back, because I think you're going to be having cool results for a long time.
Absolutely. I'm looking forward to it.
NSP6 now. I want to know what that is.
Yeah.
Yeah, and you good luck with your career. You're just getting started. It looks great, and keep it up. Okay.
Thank you. Thank you, Vincent. And thank you, Alan. Thank you, Rich. It was really nice seeing you.
Sure. Great talking to you.
Take care.
Take care.
Bye.
That's good.
Wow.
Very good. Very good.
That was great.
I have to say, when I saw the headlines, I didn't bother to investigate. And I just thought, why didn't they get permission for this? But now it's quite clear. They were following the rules. And the moral of the story is, before you make an opinion, look into the matter, okay? I'm talking to myself, too.
Yeah. Well, okay. Yeah. The internet media bubble that we have constructed over the past 20 years favors rapidly expressing a quick take. And that is not conducive to intelligent discussion.
How did we get here, Alan?
Oh gosh. I gave a like a 30-minute talk about this to a conference of science teachers.
Well, maybe it's,
in fact, I posted a transcript of the talk on my blog. But yeah, we have sort of, through a series of decisions that were mostly lazy and some of them kind of seemed to make sense at the time, we dismantled the old news system and created this new one that is now run by tech companies. But we took away all the accountability. And so, Google and Twitter and Facebook and all those can just say, oh, we didn't post any misinformation. They just provided the platform that causes people to post the misinformation, because that shows up as engagement in the statistics, because if it enrages you, you will keep scrolling. And that's the sole function of those sites.
I had
that's where we
lunch with a journalist visiting Austin who's a TWiV listener some time ago, who mourned the paucity of really science-trained journalists. And it's the phenomenon you mentioned of somebody being pulled off the sports desk to cover something because that's who's available. Okay.
Yeah, and because so even before we went to this internet model of news, the previous model of news was actually financed by, a lot of people don't really appreciate this, it was financed by the classified ads to a large extent.
Right.
And so the advent of Craigslist, probably more than any other single factor, gutted the news industry. The print news industry.
Right, so you can't hire the qualified people.
You can't hire the qualified science journalists because the big problem you have is that people don't turn to the science section and read it in the numbers that they turn to the sports section and read the front page.
It's the comics, dude.
At the comics, stories about the weather do better than stories about the water supply in Flint, Michigan. Right. I mean, like the really serious social issues, people are not interested. They had a hard day at work and they want to read something sensational and then something uplifting and then the sports scores, and, that's what sells. And so when you tie that to your business model and you now have a razor thin profit margin because your cash cow got slaughtered, you now are in a situation where you can't hire science journalists. I mean, there are a lot of us as freelancers, but most of us actually work for outlets that are more obscure, that are aimed at scientifically literate audiences. And I discovered early in my career there was almost an inverse correlation between the size of the audience and the size of the paycheck. And I said, okay, fine, I'll write for Nature and Science, and these, and I explain science to scientists a lot of the time. So
here on TWiV, you explain science to other people as well.
I do try, yes.
I have a journalist in my virology class this year, someone who's been writing, I think, mostly crime. She's covered crime mostly for the last 20 years. And I said, why are you taking this course? And she said, because science coverage by the press is abysmal.
Yes.
Cool.
You might even say it's a crime.
She is also taking the science writing course taught by Jonathan Weiner in the journalism school at Columbia.
Excellent.
And I gave 2 lectures to that last week, which I do every year. And she said, I now have 8 new TWiV fans or virology fans. So I'm doing my part. We're all doing our part here to try and fix it. It's just a slow process, and it's going to take more than us.
Yeah.
All right. I wanted to just point out one little news piece before we move on to our picks. Out of Science Insider, Chinese researchers released genomic data that could help clarify origin of COVID-19 pandemic by John Cohen. You remember last Friday we talked about the finding of animal DNA sequences in samples from the Huanan seafood market, which suggested certain animals had been infected with SARS-CoV-2, and China had removed those sequences from public servers, and now they've put them back.
Yes.
So this is good, and hopefully they can be analyzed and will shed some more light on what is going on. And as it evolves, we will let you know about it. And now let's do some picks and wrap up this episode. Rich, what do you have for us?
Well, I'm a little sorry Dixon isn't here, but I think I'll send him this link. Because
Definitely.
this is in the jazz music category. Though I have a legitimate crossover to science here, which you will see. This is an NPR article entitled "For Pianist Dan Tepfer, Improvisation is the Mother of Bach's Invention." And I just ran across this listening to NPR, as I am prone to do. And it fascinates me. So Dan Tepfer is a 41-year-old jazz pianist who I've listened to a lot of this now, and he's brilliant. He's also trained as an astrophysicist, and that becomes relevant. So he's the latter-day jazz pianist equivalent of Brian May, okay? So this is apparently, well, not apparently, in 1720, Bach wrote a series of what he called inventions. And as he was wont to do, this was for the purpose of providing little pieces for instructional purposes for one of his relatives. If you know anything about music, you may know that there are 12 key signatures. And if you consider both major and minor keys, there are 24 possible major and minor keys. And Bach wrote these small inventions for 15 out of the 24. And Dan said, I'm going to fill in the blanks. And he wrote another 9, okay? But his are, he calls them improvisations. And I would like to know his process, whether these are, you know, what his definition of improvisation is. He probably never wrote anything down. He probably, I'm guessing, played them more than once before he recorded them. But they are improvisations, and they're really interesting because they're sort of a little Bach-y and a little jazzy. All right. The article itself is really great because he explains his thinking and his process and how he relates to Bach and et cetera, and about how Bach was an improvisationist himself, and that people came from all over Europe to listen to Bach improvise. I mean, Bach was nuts. His stuff is just great. So it gives an appreciation of all of that. But I want to further point out, listen to the article because it's very good. I want to further point out there's a video clip at the bottom of the article that is a recording of what NPR calls their tiny desk concerts. And these are individual performers in an NPR studio giving a little concert. This is like 20 minutes long. And this is Dan Tepfer and his tiny desk concert. And what he's got is a player piano that will play in response to a computer. All right? And it will play as he is playing. And he's programmed this in several different ways so that the computer will, in effect, listen to what Dan is playing and improvise along with him in its own way. Not only that, but he gives his own a visual representation on a screen of what's going on at the same time. So those are fascinating. And then in the very end, he starts talking about what harmonics are and about how the intervals that are pleasing to humans are integral. Okay, so, and I forget how this goes, but a given interval, like a third or something like that, may have a ratio of 4 waves to 5 waves between the first and the third, etc. It's always integral. It's not fractions of integers. And he says, you can take these and represent them as 3-dimensional shapes. And he shows, because, this guy's a computer nerd
Because
and
he's a math
nerd,
guy, yeah.
and he shows these 3-dimensional shapes that he 3D prints, okay, that are representations of these intervals. Not only that, but of course, you can program the computer to represent these intervals on the screen as you're playing them. And he goes on to play a couple of pieces where these shapes are coming and going as he's playing these different intervals as part of the composition. Amazing. So especially if you're into music, but even if you're not, this guy is really unusual. So I would highly recommend this article.
Very cool.
Very cool.
I like that. Alan, what do you have for us?
I have a nifty video animation, 3D computer animation. It's a walkthrough or kind of a fly-through of a full 3D model reconstruction of HMS Victory, which is it's a ship of the line from the heyday of the British Empire, 18th century. It was one of the biggest and most powerful ships of its time. Huge wooden three gun deck sailing ship, and it is just a really cool tour going through, starting in the hold and what's in the hold and where these spaces are arranged and how the ship is built, and then going up deck by deck, what's on each deck, who was doing what and where the hundreds of crew members were on this ship at various, points in, like, during battle, during sailing, when sleeping, which is actually a really kind of an eye-opening. Where did they sleep? And the whole ship just fills up with hammocks, in, like, between the guns and next to each other and shoulder to shoulder, and it's just it's just a really neat way to illustrate something that you can read books about this, and you can look at pictures of it, and sometimes you can go to historic reconstructions. I've been aboard a number of different tall ship, including some historic ones like the USS Constitution in Boston, and you can climb down and see what these spaces look like. But this exploded diagram zooming through, making walls appear and disappear, and it really just kind of puts it all into a context that I think is great. It's a wonderful explanatory tool, and it's a maritime history thing, which is one of my long-standing interests, and I thought a lot of listeners might enjoy this too.
I watched this from start to finish. It's very cool.
Cool. That's very cool. Very nice.
And it'll make you it made me very glad I was never in the 18th century British Navy.
Well, and occasionally during this narration of this, he said, when the ship was engaged in action, and I thought, no.
Nope.
I don't want to go there.
I don't want to go there. Yeah, because they would be, the tactics of the day were that a ship like this from Britain and a ship like this from France, similarly equipped, would just blast cannonballs against each other. And
yeah.
Wow.
Neat. All right, my pick, it's the second one about New York. So this one is a New Yorker article by William Finnegan called "The Fight Over Penn Station and Madison Square Garden." And this was sent to me by listener Jan, and this resonates because I come into Penn Station every day. It's just 2 blocks from the incubator, and the place is a bloody mess. It is.
And it has been forever.
It's horrible. And part of the issue is that on top of it is Madison Square Garden, and the owner of that will not move it and allow the city to do renovations that it has to do. The obstacle is James Dolan, whose father is a billionaire. He established Cablevision. James Dolan was a druggie. He didn't want to do anything. His father cleaned him up and put him in the chair as the CEO. And someone asked the father, "Why'd you do that?" And he said, "Well, nobody else wanted to do it." So the guy basically owns the Garden. He owns the Knicks and the Rangers. He's apparently run both into the ground, and he has no interest in bettering the train station, which needs to be fixed. And so this is a wonderful article about the history. And I found one paragraph here that is just resonating with me. So let me find it. I have to scroll down to the end of the article. It involves politics, and this is a huge train station. Many, many people come in every day, more than anywhere else in the U.S. It needs to be fixed. They don't know what to do with the Garden on top of it. By the way, this guy, Dolan, if anyone speaks out against him, he installed facial recognition software at the Garden. If you go to the Garden, you'll get thrown out because they'll find out who you are. Oh, my gosh.
Wow. Like some other people we know. Unbelievable.
Yeah.
Yeah. So let me go to the end here, because basically they are comparing rail travel in the U.S. with Europe, where it's just amazing, right? And I had a story once about the tunnels under the Hudson River, which are a disaster as well. But here we go. Let's see. I have to find. Is it Germany? Yeah, there we go. So in Germany and Japan, track assignments. So you go to the station, you look for what track your train is, right? Germany and Japan, they put the assignments up months in advance. They publish them. At Penn, you never know what track a train will leave from till the last minute. When the announcement comes, there's the inevitable stampede for that too narrow escalator. I do this every bloody
so many memories.
night.
Oh.
I get to Penn Station. I want to take the 601. I get there at 5:58. There's no track posted yet. You wait till the last minute. This thing is called the pit, where people wait for New Jersey Transit trains. And then when they flash it up on the screens, there's a mad stampede and everybody's pushing each other. And there's a tiny stairway that goes down to the train. It's just ridiculous. And this article just beautifully describes it all. So I couldn't help but picking this one. I know it's just really specific for me, but it's a really good article. You know how the great New Yorker articles are. They just do a good job. And we have a listener pick from Douglas, who writes, "This may be of interest to you and your listeners. Thanks for your informative podcasts." He sends an article from Bloomberg. "How's your city doing? Ask the honeybees." Basically, the idea here, bees pick up all kinds of stuff, including bacteria from wherever they've been. So they're looking at bee microbiomes and seeing how they differ in different parts of New York in this particular case, and other places as well. So
this reminds me of the story. I think this may have been a TWiV pick some time ago about an urban bee colony where the bees were making red honey.
Yes, that was in, I think that, there was one like that in Brooklyn, and it turned out that they were going to the syrup at a maraschino cherry factory, which was in an open vat, because, I mean, who needs flowers if you got an open vat of syrup? And they were all loading up there, and the honey came out red as a result.
That's great. All right, that'll do it for TWiV 997. You can find the show notes at microbe.tv/TWiV. You can send your questions, comments, picks of the week to TWiV at microbe.tv. If you like what we do, consider supporting us. Microbe.tv/contribute. Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, it's really a privilege to be here. And it's always a good time.
Yeah, I mean, no matter what kind of day I have, when I sit down and do this, it kind of just pushes everything away and really This is what I enjoy doing. There's nothing better than talking good science with various
When I
people.
retired, I imagined missing what I was going to miss. And one of the things I was going to miss was just sitting around in my office yakking with people about science, okay? Which we did daily. And here we are.
Yeah,
This is
we can
great.
do it here.
And this helps organize my week.
How so?
Because week to week, I'm doing all kinds of, I'm interviewing different people about different subjects, and it's kind of come and go, all sorts of different folks. And I do get to talk about science a lot, but it's not like with a regular group.
Yeah.
Yeah.
it's cool.
And then Friday, now I get together with the group.
Alan Dove is at alandove.com, at turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.